Here we have the narrative that was used in the paper. So I'm going to take you through the whole thing. As Ben mentioned, here's the paper. There's additionally a published version of this. So this is the dynamic narrative or the interactive narrative. But there's also what's called a static narrative or a published narrative version of this that you can go and look at. And so when you do your own research in KBase and eventually want to publish it, you can create this static or published narrative from it, and it will exist for you as a separate thing that people can access without even logging into KBase. But we're going to operate in the dynamic narrative. It has basically everything winds up being here, and so since I'm trying to show you how to get through building one of these yourself, it makes more sense. So the data we're going to use for this is, let me show you the initial input data. So we have this compost enrichment from the Joint Bioenergy Institute, where they are interested in many things, but among them recovering lignocellulose degrading enzymes for biofuel production. And so in this compost enrichment, they did two enrichments, which they named 37A and 37B. And I need to get on my soapbox here for just a second and explain that how important doing different enrichments and subjecting your samples to different conditions is absolutely going to give you the best results when you're trying to recover genomes from them. So literally just take your sample if you have enough and put it in 10 or 96 or whatever number of different tubes or Falcon tubes or whatever, and then change the pH on them, change the temperature, maybe get creative, or if there's some relevant conditions with some nutrient that they need, change that amount. And so you're going to have this leads to two effects, which we'll see in this narrative. One is certain lineages are going to have better fitness for those conditions, and their relative abundance will come up in the sample. And so you'll get more reads. And so if you do the same amount of sequencing, but do it across these different conditions, these varying enrichments, you're going to get significantly better results, because in one of these conditions, you're going to have certain lineages come up, and another one, you're going to have them come up. And so you'll get reads from each of those that you wouldn't have gotten otherwise necessarily. So the other advantage we'll see later is in binning, where differential coverage binning is by far and away the best way of grouping contigs from metagenome assembly. And so you'll get a really strong signal if you have, at least 3, but, if you can do, 9 or 10 or more, it's really the best way to go. And, it's actually not that hard to do. Okay, so we're going to start out. We have 2 enrichments in this case, 37A and 37B from compost. And we are going to start by, I'm going to skip a lot of this sort of description and timing and stuff. You can go back and read it if you want. But we're going to start by taking the reads and importing them. And I'm going to skip that because I'm going to come back to it at the end. So we've got our reads in KBase as these reads objects. And data in KBase always exists as a structured object. While you can, export it in standard formats like FASTQ, or import it from standard formats like FASTQ, we store it with summary information. You can sort of see how many reads are in the object, what its GC content is, the Fred type, what the various quality scores are, and so on. And so this is very useful for operating on in a computational fashion, which is how we're going to need to access all of this. So we're going to start by importing them in, and then take a look at the quality of these data. So for example, we have this FastQC app, and we can go and we can look at the 37A forward reads, for example. And so we're going to look at the per base quality. Now, there's two things you can do in a narrative to have a sort of more real estate for looking at things. You can see this is getting cropped a little bit. One is this data pane here and this app pane here. Well, we can go ahead and just collapse those out. So we're going to do that here. The other thing we could have done is make a new tab with the report. So that will open the report in a new tab and give you the full real estate here. So our FastQC report on our imported reads is, well, they start out pretty nice, but there's really, maybe it's bleaching or something. The tail of these reads is tending to drop in quality. The y-axis here is the quality. And so this is the distribution of quality scores at that position. And we can see that the distribution really is falling off into poor quality here. So what we're going to want to do, let me close this up. Maybe I'll close this too, is we're going to want to trim these reads to keep just the high quality regions. And it's going to just mean we're operating with higher quality data. I'll skip the FastQC for the 37B library. And so what we're going to do is we're going to use, in this case, Trim-O-Matic as a quality trimming tool. And we're going to crop off, 5 from the head and make sure that each read is 140 long. So that's usually going to take some off the tail. And we can see that 96.2% of the reads survive. But in some cases, if the entire reverse read is bad, then we only get a forward read, or conversely with the reverse read, and then we're losing 0.3% of all of the pairs. And so if we do that trimming and then look at the FastQC report again for, in this case, 37A forward, we can see that, maybe I will do the separate window, that our tail is now cleaned up quite nicely. Okay, so what do we do next? Well, one thing we can do is we can try and figure out which lineages are present in these samples, just to get a sense of what's the most abundant and where we expect potentially to look for our mags to represent in lineage space. And so we're going to run this program called Kaiju. And we're going to put in the read libraries, and then we're going to select whether or not we want all taxonomic levels, and then we can pick the reference database, and then there's something we can do with whether or not we want to represent the very long tail. The population structure of microbial communities is that there's, there can be some more abundant ones, and where abundant could be even just 1% of the community. But, with 1,000 different species, for example, in a given sample, there's going to be this long, low abundance tail. And so if we start trying to plot these relative abundances, you need to start cropping that tail, or else you're just going to have 1,000 different things showing up for you. The other thing that we can do is we can do what's called subsampling. And so Kaiju is going to be running on all these reads and trying to map them to its reference database of protein sequences. And since we're really targeting the more abundant members of the community, that means we're pretty much going to get the same relative abundance if we look at, say, 10% of all the reads instead of all of them. And that's going to save us a lot of time in the run. So for example, if we look at the job status, this Kaiju run only took an hour and 44 minutes, whereas if we'd run on all of the samples, it maybe would have been closer to, say, 10 hours. And we get pretty much the same results as if we had done all of them. This is not something that Kaiju offers by itself. This is something that KBase we added as a way of speeding it up. So here we've got our 37A and 37B samples, and I'm going to jump to a separate tab. And so we can see that the fraction of reads that are classifying are 40% and 50% pretty much. And so, what's going on in this unclassified fraction? Well, they may represent novel lineages, or they may represent just, garbage sequence. There's just always some weird sequence that shows up. And so, it's not clear what's in the unclassified fraction, but we can at least look at the lineage assignments for the classified reads. It may also be that these are reads that are too short to have a protein sequence that matches the reference above some confidence threshold. So novel lineage, or just simply the read was too short. We're talking about Illumina paired end reads here that we've now cropped to 140 base pairs. So, a lot of times that doesn't necessarily cross a confident protein. So we're getting a lot of proteobacteria, we're getting a lot of bacteroidetes, and so on at the phylum level. And we can zoom down and it starts to fragment more, as you come down. So order, family, and so on. And so we're going to, as you can see, this is why you want to have this tail region in the report, so that you don't see it gets longer and longer. Imagine if we hadn't cropped it at 0.1%. And you can, when you go to look at the report, there are, in fact, files you can download that will include the unbucketed tail. So you will be able to get that information for the download. It's just not in a table. You just can't see it in the plot. Okay, so it looks like we've got some similar lineages in the more abundant fractions in both 37A and 37B, but they are in different relative abundances, and especially down here. And so we're going to go ahead and do a metagenome assembly. We have several different metagenome assemblers on KBase. One of them is Metaspades. And so we're going to start with Metaspades, and we're going to do an assembly. And you get a quast report from that assembly. And so it'll have an accumulation of sequence, plot, and so on. But let's sort of skip that for now, because we're going to come back to it. So we do a 37A assembly by itself. We do a 37B assembly by itself. And then we also, since we saw that there were lineages that appeared to be related in both 37A and 37B, we're going to merge the read libraries as well. And this gives us a merged read library, which we're going to call 37AB. And now we're going to assemble the 37AB as well. Here it is. It's our input. One thing to note is metagenome assemblies. These assemblers will produce, any joining of reads that they can. But like with lineage abundance, you also get this very long tail where most of the many of the contigs, I shouldn't necessarily say most of them, but often most of them are very short. And so a lot of what we're doing downstream of this is going to be trying to group these contigs into one lineage, a process called binning. And a lot of the term used there is k-mers. And so very short contigs aren't going to have a very strong k-mer signal. The other thing is if you're looking for proteins in these contigs, well, if you have, a 500 base pair contig, maybe you'll perfectly fit a protein in there, but a lot of times proteins are longer than that. And so you're going to really wind up having, lots and lots of contigs. It's going to slow down your analysis, and the value of those shorter contigs is significantly less than the longer ones, especially if you're trying to get at things like genome context and looking at operon structure and so on. So what we're going to do here is we're going to cap or put a floor on our contig length of 2,000 base pairs so that we wind up with really just sort of the higher quality contigs in our assembly. So we run that, and now we have the assembly from that. And again, we're going to skip the quast report here because looking at it by itself is not as meaningful. And then we also are going to do assemblies with megahit, since we have that, and then we're going to do the co-assembly there. We're going to do it with both the meta large, where you expect you have a preset set of parameters for tuned to a complex community. We're going to do meta sensitive, where it's going to put more work into assembling the more abundant members. And then we also have IDBA-UD, which is a method optimized for variable population structure in your community. And the fact is, we don't know going in which of these assemblers is going to produce a better result. So we're trying a few of them. You may know which one you prefer, or you've used one or two of them on your data. And so you don't necessarily have to go in and use all of these assemblers. But this is for sort of the illustration of variation that you get out. So now we're going to compare these contigs that we get from each of these assemblers. And here's a table that does so. But we also have an app, which is very much like Quast. But I found Quast was missing a few things that I wanted, like histograms of the lengths. So I added those in this app, compare assembled contig distributions. So we retained the Quast-like cumulative length plots and sorted contig length plots. And you can see here that the co-assembly with metaspades produces the greatest proportion of long contigs. And as I mentioned, I'm going to open in a separate tab. As I mentioned before, I wanted to be able to look at the length histograms. So I created this app, and you can see that there are more long contigs, even up to 1.4 megabases, in the 37AB metaspades assembly. And these other, megahit and IDBA, they're doing a pretty good job too. But you can see here that the larger proportion of the distribution is occurring for the 37AB metaspades contigs. And this isn't necessarily valid in terms of metaspades tends to optimize joins with less support, let's say, than the other methods. So it's possible that, we're getting false positive joins just for the sake of having longer contigs, but maybe it's getting, because it's doing more joins, some of them are true positives as well. So it's like anything in bioinformatics, there's a balance between sensitivity and specificity. And in this case, we're going to lean towards sensitivity. But sometimes maybe depending on what you're doing, you want more specificity. So, okay, so now we have our assembled contigs, and we're just going to move forward with the co-assembly using metaspades. in this case. And we're going to start and use max bin 2 as one of the binners we have. So with this, as I mentioned, having variation in your samples is very strong signal for binners, which these days all use relative abundance as one of the strong signals for trying to group the contigs into one lineage genome or bin. So what we're going to do is we're going to take our co-assembled contigs, and then we're going to have the different separate read libraries as the input. So 37A by itself, 37B by itself, and we're going to produce a binned contig object as the output. And so we run the binner and we get 68 bins, and 82% of the contigs are binning, 18% are not, and so on. But then by itself, a given binner is only so reliable. So what we're going to do is we're going to also run metabat, which is a binner that uses Kamer frequencies and relative abundance as well, but has a slightly different parameterization from max bin 2, and it's going to produce its own set of bins, in this case 76. And then we're also going to run concoct, and concoct makes 106 bins. But by each which of them is right? Well, none of them. Can we improve on them? I should say none of them are perfectly right. So can we improve on what they've got by building a consensus? And so I'm sure most of you have heard of Jill Banfield. And so out of her lab, they developed something called DOS tool, which takes the input from different binners. In this case, are max bin 2, metabat 2, and concoct bin assignments for these contigs, and it takes in the contigs themselves and then tries to build a consensus set of bins out of the input bins. And so if we look at the results, so in this case, the evaluation of the bins is done with single copy universal genes. And so these are typically ribosomal proteins, but there are others that are used as well, like RNA polymerase B and so on, gyrase, DNA gyrase, and so on, where it's a essential gene for microbes, but if you wind up duplicating it and you have divergence in it, that can kill the cell and become toxic. So only one copy is maintained. And so because these are such important genes, they can be used you can make the statement that they are almost always present in a single copy in a genome. And so you can evaluate the quality of genomes without knowing its lineage at all by how many of these single copy universal genes are present in it. And so that's what's being done here, is how many of these are present in a given bin. So here are the inputs, max bin 2, metabat, and concoct. And here's the DOS tool bin set. And you can't really see, but there are more of the bins that are more complete. And it's easier to see with this report, where you have max bin 2, metabat 2, concoct, and then the DOS tool, where the dark blue here are the bins that are greater than 90% complete by single copy universal, or single copy gene is what they're calling it, but it's also universal. More of the bins are more complete. And so at high quality, medium quality, and so on, you just have better quality. So we're going to take the DOS tool consensus bins as our bin set, and that's 40 bins here. And now we're going to assess the quality, not by just the single copy universals, but we can use a tool called Checkm. And there's a newer version that's coming out. Well, it's out now, but it hasn't fully been released, called Checkm 2. We don't have Checkm 2 on system yet, but we will once Checkm 2 is sort of fully released and published. So in the meantime, we're going to use Checkm. And what Checkm does is it takes the bin and looks through not just for the single copy universal genes, but it does a phylogenetic placement of the bin genome into its species tree. And the species tree allows you to calculate additional genes that are single copy markers for that lineage. So for example, maybe you are talking about desulfovibrio, and if you place a bin in desulfovibrio, you know that all of the sulfate reducing machinery also is going to be present. Maybe not a single copy, but there will be collections of genes that are, essential for that lineage to function. And so you can expand the single copy markers into lineage specific or clade specific marker sets. And so that's what Checkm is doing, is it starts by taking your input binned contigs. So I've, used the name of the object to sort of capture the history of it. And it's too much detail to go into here, but we have a system on KBase called Provenance, where you can actually track the data objects and the apps that have been run on them to produce that a given object, like this binned contig object. But just for my ability to track it without resorting to looking at the provenance graph, I embed a bunch of information in the name, that it's the co-assembly from Metaspades, and that these are the 3 binners that went into DOS-tool to produce the bin contigs. So we're going to go and run Checkm. You have a choice of either the full species tree or a reduced tree. And we're going to save all the plots that Checkm makes so we can download them. So here we have the Checkm plot. So we have all of our bins, and they are being scored by how many of the clade specific markers they have. So here you can see they're quite a bit more than, say 40 or 50. There's a very varying number of what is considered a single copy universal gene, or bacterial universal or archaeal universal. But typically it's on the order of 50. So we're getting more than that here. And so we're getting representation of a bunch of the markers for bin 30, but a lot of the others are missing. That's what these grays are. And then for some of these lineages, like bin 1, for example, there are actually quite a few markers that are being found. And so you wind up with the plot having very, very fine lines here. And sometimes if it's too small, it'll actually just be very, very hard to see the separation. Anyway, so if we go down to the, like this bin 20, for example. So if we go down to the bottom here, there's the key to this plot, and that is green means that there's a single copy of that clade specific marker, gray is missing, and then we have this additional 2 colors, blues and orange reds, and that is meant to indicate, in the case of blue, when there's more than 1 copy of that gene family showing up in the genome. And if it winds up being close to or if the multiple copies wind up being close to one another, then you can say it's probably a paralogous expansion of that gene family in that lineage. It still may represent strain variants genomes that are being mixed, but it's hard to distinguish that from a paralogous expansion in the lineage that's legitimate. And so those are these, they're light blue in this, say, bin 47. Contamination is when you have more than one copy, but the two sequences are distal to one another, and those are more likely contamination. In this case, not contamination of the sample, but the bins themselves have had their contigs mixed. And it, maybe it's correct and maybe it's not, but overall, you can see that. Why can I not scroll? There we go. That the best of the bins are here at the bottom of this plot, and the worst quality ones are here at the top. We also offer a table where you can look at the completeness and contamination of each of these bins numerically. And again, one has to take these numbers with a grain of salt, because if it's a well-studied lineage, then yeah, maybe the clade-specific markers are valid. But then you have other lineages like Putative bacteria, where it's the knowledge of what are the clade-specific markers is less well in hand, and so it may wind up with a completeness score that's less than, say, 90%, even if it's a complete genome. So these are approximate guidelines. So what we're going to do following this is we're going to filter out the lower quality bins, and we're going to, because we have so many really high quality bins from this, again, enrichments are a good way to go. We are able to threshold at 95% completeness and 2% contamination, and so we're going to do that to find the really, really best quality ones. And we've made a variation of the check-a-map where we filter the bins for you in this app. And so it'll produce a bin contigs object as its output that is just the high quality 95.2. And here I need to make a quick statement, and that is what we're calling high quality here is based just on the Check-em completeness and contamination scores. Metagenome-assembled genomes from short read sequences like we have here, Illumina, for example, often have difficulty assembling ribosomal RNA genes. This is because they're often present in more than one copy in a genome, and therefore the assemblers get confused and don't wind up being able to span repeats. This is also true for longer transposons, where there can be multiple copies that are identical or virtually identical in the sequence. And so this breaks assemblers' abilities to join those shorter contigs as they're doing the assembly. So here we wind up filtering out those short ones. Sorry, it still reports all of them. But if you look at the table, you'll see in red here what was below our threshold. So, here this bin 4 actually is pretty good quality, but it's below our very strict 95.2% threshold. And so it gets filtered out from that bin contig. And then we're also going to do one at 90 and 90% completeness and 5% contamination, just to capture those that are sort of in the middle. And so we'll have a set of bin contigs there. So now what we need to do is everything is stored as a bin contig object, but what we need to do is pull out each of those as a separate assembly representing that lineage. And so we're going to run this extract bins as assemblies method, and we're going to run it, say, in this case, on the 90% completeness, 5% high quality fraction. And it's going to pull those out, and it's going to produce assembly objects for us. and it will also create what's called an assembly set. And this is a nice object to have, because now instead of having to add each of these assemblies at once separately to apps that are downstream, we can now just give it the assembly set that holds them inside. And that means that it's much easier to run these apps. So for example, after we have these assemblies, well, now we need gene calls on them. So we can run annotate multiple microbial assemblies, this RAST app, but instead of having to input each one of these assemblies one by one, which you could do if you had a certain subset that you wanted to work with, we can instead give it this 95 assembly set as its input, and it will produce a genome set as its output. So it will produce genomes, which are assemblies plus gene calls plus annotation, functional annotation, and additionally the genome set that we can use as a single object input for other apps. So now we're going to try and get a better handle on what the lineages are for these mags. So now that we've pulled them out of the bin contigs, and we decided that these are going to be, these are the high quality ones, we're going to, go forward with analyzing them like they were similar to isolate genomes. We're going to start calling them mags now, because they have gene calls to them. They really do look and can be operated on like regular isolate genomes. So now we're going to use this method called GTDBtk. So GTDB is a whole genome based alternative for taxonomic classification. And so what the Hugenholtz group has done is they have taken all of the mostly high, but some medium quality genomes, isolates, and mags from NCBI and built whole genome species trees using the single copy universal markers for tree building. And they can then use that to go and place new genomes, new mags into that taxonomic classification scheme. And so that's what GTDBtk does. It does two things. One, it runs fast ANI for a quick assignment, but then it also does a phylogenetic placement in the tree using pplacer. And so you'll wind up being able to, for those genomes that can be classified, you'll get some classification in the GTDB taxonomic space. In this case, bin3 is able to be classified to this genus, but not to a species, whereas others, at least not on this first page. Here, bin37 winds up actually being within the similarity thresholds to be put into this species classification. So you'll get a different range of it. Some of them will be closer down to the species level, and others will be just say to the family level or so on. So now we have these, and we've updated the genome object to contain this taxonomic classification. Now we also want to look at these within the species tree context. And so we're going to take our bins and we're going to put them into a species tree against refseq genomes using the species, this insert set of genomes into species tree app. Now, the species tree app has a tendency to just find the closest genomes. And so you can wind up with a situation where lots of genomes are going to be found for certain lineages, but then others, so for example, this bin 43 here is finding lots of these lineages. They're just well represented and close to this bin in RefSeq, whereas some of these other bins are not finding the closest relatives. And so what we need to do is split up our bins into different clades so that we don't wind up having, examples like bin 43 sort of dominating which species show up in the species tree. So we're going to split them up into clades, and we're going to do this manually using this build GenomeSet, where we specify, no, those are missing. Specify that clade A is going to be bins 3, 42, 57, and 59, which is what is supposed to be showing up here. And that's based on, well, I'm not, oh, here we go. Yeah, 59, 3, 57, 42. So that's clade A. And we're going to do that for clades A through K. So this is just by eye looking at the species tree that was generated with that first run. And then we're going to go and insert each of those clades separately into the species tree to find the proximal genomes. So here's all the build GenomeSet runs until we have K. And then we're going to insert, in this case, clade A into the species tree. And we're going to add 20 RefSeq genomes into that tree. And it's going to give us more proximals RefSeq genomes. Here's clade B, and so on. So anyway, this is a way of sort of allowing us to figure out what are the close relatives to our bins. And potentially we want to include them in downstream analyses of, say, function or pan-genome analysis or something like that. That type of downstream comparative genomics, we're not going to have time to discuss today, but early in 2023, I'll do another webinar where we go through that whole process. So we're going to go and finish inserting our clades into the species tree and finding all the RefSeq proximal genomes. There are a bunch of apps runs here. Additionally, maybe we don't want just the proximal RefSeq genomes, but we want to have a sort of bigger picture phylogenetic context. And so we can build a species tree where we have genomes that represent a given phylum that are sort of forced to be in the tree. And so that this build microbial species tree app allows you to do that. So we have our mags with, we're going to use as a phylogenetic skeleton, the bacteria, since we know we don't really have any archaea in this set of mags. But you could use bacteria plus archaea, isolates, mags, and sags. You could use just bacteria isolates and so on. Here we're going to do sort of all bacteria, mags, and sags. And we're going to have our phylum skeleton species tree. And now we can look at the bins in this phylogenetic, super context, or the phylum context, I should say. And I'm going to zoom in a bit on this. So here we can see these bins are showing up in alpha proteobacteria. These bins are showing up in vericomicrobia, and so on. Here's our Bacteroidetes sclerobi. So that's another sort of phylogenetic app we can use. And now we want to look at the functional classification of these mags. And we have several different tools for this as well. So, we ran RAST, and that can give us seed classification. And there's tools for looking at the functional profile of seed. You can run COG or PFAM or TigerFAM's annotations on these genomes, and you can look at the functional profile those domain classifications. Here, I'm going to talk about a little bit about the DRAM functional distillation app, where we put in our assembly set, and we're going to get genome annotations using DRAM out of that. And additionally, DRAM provides this really nice table. Sorry, these are all the created objects here at the top. Can collapse that. And instead look at the DRAM report. And so here we have each of the bins as rows, and then these different DRAM functional categories as columns. And so you can have electron transport chain classification. There's these different glycolysis modules and so on. And then additionally, for metabolisms, DRAM offers KZ classifications, nitrogen metabolism, and so on. So that's one route towards functional classification of your bins. We additionally can use direct hammer profiling. We have several different suites of gene families available, and are expanding those different sets. Right here, I'm explaining, I'll tell you about the KZ family, since this is of particular interest for this study. The DBCAN group has taken the KZ families and built HMMs out of them. And so we can run our bins through that, and we can get the GH1s, GH2s, and so on. And one nice thing about the Hammer app is we can, when we configure it, we put in our GenomeSet, and we can pick different families, but we can actually specify particular families, like say GH4, and then we can choose to save feature sets for explicitly requested models. You can save it to all models, but then you wind up with a ton of feature sets, one per gene family. So in this case, we're going to save only hits, or we could save only hits to explicitly requested models, like, so for example, like I said, GH4 or something. And that would produce a feature set that we could then use in subsequent analysis. Again, I don't have time to go into that today. One of the collections of gene families we offer, HMMs we offer, is called microtrait. And we can use microtrait in this summarized GenomeSet app, where to give a sort of functional summary classification for each of these mags. And then additionally, the summarized GenomeSet can run check'em for you. It can add this microtrait bio element. You can choose to show just the newest genome version as you've updated it over and over and so on. And you can use there's different parameters for check'em and so on that you can include. So since I'm tight on time, let me just keep going forward here. And we have our bins, and we have these sort of summaries of the functional categories, and we can see that bin 3 doesn't seem to have any of the nitrogen fixation genes, but for oxygen tolerance and utilization, we have these genes families showing up. There's cis-N and so on. Bin 11 is showing up, bringing in SOX-B, SOX-Y, and so on. So we have the ability in the summarized GenomeSet app to have the genome, the mag, it's a functional summary and what genes are present in it. And this is downloadable as a table too, so you don't have to just eyeball the rollover. The taxonomy of it, its check'em completeness, its contamination, how many contigs are in the mag, the genome size if you sum the sequence of those contigs, its N50, its GC, how many coding sequences there are, CDSs, how many tRNAs are present. And this is, as I mentioned, A lot of MAGs don't wind up getting the ribosomal RNAs assembled because of the fact that they're repeats, and this is a shorter read technology. There are long read technologies coming online for metagenome assembly that resolve this problem, and so it's really probably best to use a combination of the two. But we are getting CRISPR arrays and so on. So there's this summary does show up, and in fact, sometimes you do get a 16S here, and so it's still not going to meet the MeMag threshold for isolate level quality, but bin37 appears too, and so on. So you can use the summarized genome set to sort of have a look at everything that's coming out of your MAG recovery. So we'll be, like I said, in early 2023, be doing a comparative metagenomics and MAG analysis tutorial. There's a bunch of reference literature here. And let me go back up to the top, since I'm just about out of time. This narrative, as you can see, it's chock full of data already. And you may want to be going through this analysis with your own data. And this is, going to be in the way, and all these app cells are already here. And so what we've done is produced a blank narrative or a reset narrative or a template where the data has been removed, the app runs have been removed. And so what you want to do is go to that narrative, and then copy it. Maybe you want to wait a little bit so we don't all wind up doing it at the same time, and because this is hard for the system. You go to this template narrative, click on narratives here at the top. Well, it's a little slow. Anyway, then you would click on copy this narrative, give it, a new name like my metagenome narrative, version 1. Copy it, because you're not going to be able to edit this template one. You're going to want to make a copy of it so that it's your copy and you can work on it, and it's your private narrative and so on. So now I'm going to open that up. And then this is the one you're going to want to import your data to, and all of that. So we're out of time. And so I'm going to very quickly see if there are any questions that haven't been addressed.
So thanks again to Dylan and the whole KBase team for all the work that went into this paper and the narratives associated with it. And we hope to see you all next year on more of these KBase webinars. And of course, we hope to see you on the system. And I look forward to all the papers that you will have coming out using this pipeline. So, okay, take it easy, everyone. And we'll post this recording up tomorrow or so on our YouTube page. So thanks again.
Thanks for watching this webinar. For more webinars and tutorials, subscribe to our YouTube channel. You can see announcements for upcoming webinars and recordings for previous webinars on our website at kbase.us/learn. Let us know in the comments what content you'd like to see in future webinars. Be sure to follow us on Twitter at DOE_KBase. And if you have questions or encounter an error when using KBase, please contact the help desk.